Hello, everyone, and welcome to the last live session of the first International Electronic Conference on Genes from this year. This conference has been organized by MDPI and MDPI Genes Journal, and is being chaired by Dr. Aureliano Bombarelli from the University of Milan in Italy. As always, I would like to thank our chair and the conference committee for making this conference possible, and all the participants for joining us today. Until the end of the month, everyone can still read and comment all the author submissions presented for the conference. So please look at our webpage for more information. I would also like to remind everyone that this webinar is being recorded and that we have a YouTube live streaming. The recording will be posted on the conference webpage for everyone to watch it as soon as possible, and we will be sharing the YouTube link on the chat. Due to some technical problems, the speech of Dr. Simone Rampelli is pre-recorded, but he will be connected during the whole session, and at the end of the session, he will answer some questions. So please feel free to post them in the QA box at any time. And if you would like to ask your questions on live, use the raise hand option, and we will upgrade you as a panelist for a moment, letting you have your camera and microphone on for asking your question. If anyone needs anything during the session, please write us the panelist using the chat option. Finally, I would like to present the speaker for this session, that is Dr. Simone Rampelli. Dr. Simone is an assistant professor in chemistry and biotechnology of fermentation from the University of Bologna in Italy. His research activity, documented by 55 publications in international peer-reviewed journals and more than 30 participation in national and international congresses, is mainly focused on the characterization of the compositional and functional structure of the human gut microbiome and its impact on human health. Thank you, Simone, for accepting to participate in the session. I will start now in a moment the recorded speech, so everyone enjoy.
Good morning, everybody. I am Simone Rampelli. I work as assistant professor in metagenomics at the University of Bologna. During my career, I got the opportunity of working on the microbiome of extreme long-lived individuals, such as centenarians and semi-supercentenarians. That means individuals that join the extreme limit of the life with more than 105 years old. But let me introduce the topic with some slides, and after I will show you something about my research. We born sterile and we develop a stable microbiome at higher level of biodiversity only after weaning, and we maintain such configuration for all the adulthood until a subject-specific threshold in elderly. After that, microbiome shifts to configuration at lower level of biodiversity in old age. So, as I said you before, one of my research interests is focused on the characterization of this last part of the trajectory of the human microbiome. In particular, we try to address the following question. When does the gut microbiome start to change in healthy elderly people? Are the occurring changes to be considered dysbiosis or adaptation to age condition? Here I put a list of studies investigating aging and the gut microbiome in humans. As you can see, there are not a huge number of studies, probably because it is not easy to find samples from very old individuals. However, here there are a list that ended in 2017. There are other work, but they are not so numerous, as you can see. Overall, these studies say that the physical changes and physiological changes occur in the human host along with aging, that include the deterioration, nutrient intake, and absorption, including also problem in dentition, probing salivary function and intestinal transit times, as soon as sensory changes in taste and smell, physical changes to the intestinal epithelial barrier, inflammation, and immunosenescence. All these factors affect the gut microbiome. In particular, the H-type microbiota is characterized by a low biodiversity, an increase in pro-inflammatory pathogens, such as Enterobacteriaceae, Staphylococcus, and Streptococcus, a reduction in immunomodulatory groups, such as bacterial member of Clostridium cluster 4, and a reduction in short-chain fatty acid producer, such as member of Clostridium cluster 14A. Claassen and colleagues in 2012 gave a huge contribution to the field of healthy aging. In particular, they enrolled 178 individuals from different settings, including community, hospital, and short-term or long-term residential care. They collect a sample from this individual. They characterize this the sample, so the microbiome, by means of 16S RNA sequencing. The study was not completely focused on healthy aging because they did not enroll very old subjects such as centenarians. However, they focus on the impact of lifestyle determinants on microbiome that are quite important aspects for healthy aging in elderly. In particular, they found that community-dwelling elderly people show a microbiota structure overall comparable to that of young adults. Furthermore, they found that elderly people living in long-term residential care show a more compromised microbiota structure. And this compromised microbiota structure was associated to the dietary changes occurring along with age, to an increased score in frailty and related factors, to an higher inflammation, a lower production of short-chain fatty acid, hospitalization, and immunosenescence. More recently, we study and characterize the microbiome centenarian and semi-supercentenarians as the best model of healthy, successful aging and longevity. Essentially, these are individuals that were more than 100 years old, and they show a low incidence on chronic illness, a reduced morbidity. And in light of the fact that they are very old, we can say, all things considered, that they are healthy. We found a core microbiota that is shared among young adults of 30 years old, elderly people of 70 years old, centenarian and semi-supercentenarian, that comprise highly occurring symbiotic bacterial groups, such as Coprococcus, Roseburia, and Faecalibacterium. And this core microbiome remains approximately constant during aging, but varies in the cumulative relative abundance of its member along with age. Furthermore, aging is characterized by an increased contribution of subdominant bacteria, such as member of Enterobacteriaceae and the Sulfovibronaceae family, as well as a rearrangement in their co-occurrence network. Finally, the microbial ecosystem found in extremely old people is invaded by microbes typical of other niches, such as Mojibacteriaceae, and enriched in the health-associated taxa Akkermansia, Bifidobacterium, and Christensenella. And here, just to focus on these three last genera and family. So Bifidobacterium is a well-known probiotic agent with anti-inflammatory and immunomodulatory properties, a long history of use. Akkermansia is a mucin degrading bacterium recently proposed as a next generation probiotics. And Christensenellaceae include the genera Christensenella that was recently proposed as next generation probiotics specifically developed for healthy aging. It is important to note that both Akkermans and Cristesenella harbor genes coding for deglicases, which alleviate the aging-related burden of advanced glycation end product. And these deglicases are quite relevant because are connected to this advanced glycation end product that are connected with the end aging project, and an increase of this molecule was also associated to other chronic disease, in particular with vascular problems. So overall, we can say that healthy aging is characterized by an establishment of a new homeostasis within microbiome and the host that contribute to the maintenance of health. Our research at this point focus on two aspects: taxonomic characterization of the microbiome species level and a functional characterization of the microbiome in healthy aging and in extreme longevity. So we have the opportunity to explore this aspect because we get the access to a very peculiar cohort. In this cohort, we have several different groups of individuals. Well, young adults of more or less 30 years old, elderly people 70 years old, centenarians, and semi-super centenarians as individuals that were more than 104 years old. So we get fecal sample from the subject. We extract DNA, microbial DNA, and we prepare the library for shotgun sequencing by using this kit, Kiasec FX DNA library kit, and for obtaining 450 BP fragments, and we sequence this fragment with Illumina machine, in this case, NextSeq Illumina machine, to run for obtaining 370 gigabases for the analysis. So we obtain with analysis evidence of an age-related trajectory involving the establishment of age group-specific topological pattern in the taxonomic and functional microbiome structure. In particular, we first focus on the species level characterization. We found a high individual variability of the gut microbiome, with only 3 species that were detected more than 0.1% of relative abundance in at least 70% of the sample. These 3 species are Faecalibacterium prausnitzii, Bifidobacterium bifidum, and Escherichia coli. When we look for difference among the age group, we found that Bacteroides uniformis, Eubacterium rectale, Faecalibacterium prausnitzii, and Coprococcus comes characterize young adults and elderly, 70 years old elderly people. On the other hand, we found that Egertella lenta, Akkermansia mucinifila, Escherichia coli, and Methanobrevibacter smithii characterize the microbiome of centenarian and semi-super centenarian. After that, we focus our attention on the metabolic potential of the microbiome of the specific age group, and we found that semi-super centenarian and centenarian show distinct metabolic potential in the microbiome. In particular, they show a decrease in gene for carbohydrate degradation. Here you can see on the right that there is a sort of decrease for all the genes that are involved in starch and sucrose metabolism. In parallel, we also found increase of gene for tryptophan metabolism, and difference in also are detected in the pathway that are involved in lipid metabolism, with an increase of alpha linoleic acid metabolism and glycerolipid metabolism in centenary and semi-supercentenary respect to younger individuals, and a decrease of glycerophospholipid metabolism and sphingolipid metabolism in centenary, semi-centenary, always respect to younger individuals. We also found another interesting aspect. So in this case, an age-related increase in the number of reads for gene devoted to xenobiotic biodegradation and metabolism. So centenarian and semi-supercentenarian show higher reads assigned to gene involving toluene degradation, chlorocyclohexane and chlorobenzene degradation, ethylbenzene degradation, and caprolactam degradation. This is quite interesting because toluene, ethylbenzene, chlorocyclohexane, and chlorobenzene are quite important for human health. Indeed, the primary man-made source of this molecule are the emission from motors and exhaust vehicles, as well as cigarette smoke. Furthermore, they are known to be generated during the processing of refined petroleum products, such as plastic, and to be contained in common consumer products, such as paints and rubber. On the other hand, caprolactam is the raw material of nylon used for the production of many indoor products, such as synthetic fiber, resins, and plasticizer. Well, previous study and demonstrated that higher indoor burden on the xenobiotic molecules and emphasize the exceptional importance of indoor exposure on human health. Furthermore, it is a matter of the fact that a live environment with a strong anthropic pressure, such as that of Emilia-Romagna region, that is the region where subject role in the study life, bring to a continuous and constant exposure to the pervasive xenobiotic substance that I mentioned before. So when we raise the question in the top of the slide, why do centenarian and semi-centenarian show higher metabolic potential for xenobiotic biodegradation and metabolism, we take into consideration that in our case, we have 100 years old people and semi-centenarian that are older than the other subject, and just for this reason, they show a very important higher exposure due to their longer life to this pervasive molecule. On the other hand, they also showed a reduced mobility, so this means that they spend more time at home. However, we have not monitored xenobiotics in feces and body fluid. Taking into consideration all this stuff, we also raise an hypothesis that this age-related variation is adaptive. That means that this exposure could create the appropriate condition for the human host to select for gut microbiome components capable of detoxifying such chemicals compounds with a mutual benefit in terms of microbiome and host fitness in anthropic environment. To verify that, we sought for the proportion of reads for xenobiotic degradation assigned to gut commensal on total reads assigned to xenobiotic degradation. We speculate that the highest contribution that we found in semi-supercentenarian and centenarian to xenobiotic degradation to commensal in long life people in this long life may be the result of a top-down selection process related to the lifestyle habits of centenarian and semi-supercentenarian that I mentioned before. So more time at home and lifelong exposure. After that, we finally looked to resistance, starting from age-related variation in the taxonomic composition of the human gut resistance. And we found that the taxonomic structure of the resistance largely overlapped with that of the microbiome, as antibiotic-resistant genes are mainly arbored by the dominant family of the gut microbial ecosystem. As you can see here, we have a higher abundance of reads for antibiotic-resistant genes assigned to Bacteroideaceae, Eubacteriaceae, Prevotellacea, Eubacterium, Faecalibacterium, Roseburia, Ruminococcus in young adults and 70 years old elderly people. On the other hand, we have an higher abundance of reads assigned to Enterobacteriaceae and Deggertella in centenarian and semi-supercentenarian. And when we look to the function, we also found difference among the age group for targeted molecule antibiotics, particular centenarian, semi-supercentenarian show more gene that target the sulfaminomide, rifampin, and tetracycline, along with more multi-drug gene, multi-drug channel. Young adults show more gene for the confer resistance for beta-lactam antibiotics, and the elderly for macrolide, lincosamine, and streptogranine. So we can say that as before for xenobiotic, we can observe that we can advance this hypothesis that the transition to a new resistant configuration could be the result of a top-down selection process connected not only to health status and past medical history, but also in this case could be connected to the lifestyle habits that include a stable and constant living setting within home and longer and more extensive exposure to various chemicals. So to summarize and to conclude, we can say that the peculiarities of extreme aging gut microbiome structure are connected to adaptation to physical, lifestyle, and physiological changes of the human host. In other words, for example, the rearrangement of bacterial species that include an increase of the pathogens Escherichia coli, a decrease of the short-chain fatty acids producer Faecalibacterium prausnitzii, and an increase of Methanobrevibacter smithii, together with an increase in health-associated taxa such as Akkermansia, Bifidobacterium, and Christianellaceae, are associated to the increase of host inflammation, immune activation, and changes in diet. Secondly, the higher proteolytic metabolism that include evidence of higher metabolic potential in tryptophan degradation is connected to an increase of branched-chain fatty acid production in intestine and a connection with immune activation. The lower saccharolytic metabolism that include the reduction of the metabolic potential for starch, sucrose, and glucose degradation is associated to a decrease of short-chain fatty acid production. The rearrangement of lipidic metabolism that include an increase of rates for alpha-linoleic acid and glycerolipid metabolism pathway, and a decrease for sphingolipid and glycerophospholipid pathways in centenary and semi-centenary is possibly reflect the higher intake of plant-derived fats than animal ones. The higher xenobiotic metabolism might be connected to a long life exposure to this molecule and the reduced mobility of centenary, semi-supercentenary respect to the younger subjects, and the resistant rearrange and increase of antibiotic-resistant genes, above all in pathobionts, raise attention to the progressive age-rate accumulation of antibiotic-resistant genes with potentially severe repercussions on health aging. So to conclude, we can add the gut microbiome as a new target for activating major host longevity signaling and promote health aging. Particularly, we can target diet by increasing the consumption of dietary fiber for increased abundance of health-associated taxa and high level of short-chain fatty acid production, calorie restriction for a more efficient metabolism, lifestyle with physical activity that can impact on some taxa, but also outdoor life, increase outdoor life for xenobiotic and antibiotic-resistant exposures for decrease xenobiotic and antibiotic-resistant exposure. After that, also we can also foresee the administration of traditional or next generation probiotics, for example, bifidobacteria, or some new next generation probiotics such as Greensteinella or Faecalibacterium prausnitzii, when lacking in the gut microbiome structure. Fecal microbial transplantation could be another direct means to manipulate the microbiota if necessary. We can also foresee postbiotic administration, that means directly some bacterial products, or phage therapy for example, pathobionts could be a strategy if necessary. If allowed. Well, this is my group, and these are the people that are involved in the research. I have to thank in particular Elena Biagi, Silvia Turroni, and Professor Marco Candela and Professor Patrizia Brigidi for the work, and also Teresa Tabella for the part on resistant genes, and all the other people for the collaboration, for collaborating on this topic. And this is my contact email, and thank you very much for the attention.
Okay, so we have finished with the video prepared from Simone Rampelli. So if anyone wants to ask any question, okay, so we have the first question. Given the high diversity found in the gut microbiome, would you say that certain microbial combination in our gut help aging in a better way?
Yes, I mean that, because we know that there are some families in our gut that are, such as pathobionts, that are in some way nurturing inflammation that is a sort of characteristic of aging, of the aging process, So if we can, in some way, contribute to deplete this part of the microbiome, and on the other hand, give some good bacteria, some bacteria that can promote the health of the intestine in some way, such as short-chain fatty acid producer. This is better, of course.
So while we are waiting, oh, okay, we have another question. I was going to ask one. About the xenobiotic compound biodegradation. Apart from your studies in elder people, have you thought about doing gut microbiome studies comparing the prevalence of these microorganism species in people living in urban areas and in the countryside?
We perform that kind of study when we have the opportunity of studying the other hunter-gatherers from Tanzania. So some people that live very away from the city, very away from industrialization and all the typical environment and pollutant that characterize our Western country, and we found some difference, and so they show less gene in their microbiome for the biodegradation of xenobiotic molecules such as benzene and toluene molecules. Respect to Italian people, the microbiome of Italian people, yeah.
Okay. Before going to the question of Peter, because we have another question that I think is really related to this one, were all the participants from the same city region?
Yeah, they come from all from Emilia-Romagna region, yes. In Italy, yeah.
Okay. So, Peter, if you want to ask your question.
Hello, thank you very much. That was a really interesting talk.
Thank you.
Of course, it's very interesting to find out that there are these differences in different age groups, and particularly in these elderly people. But there are many things that are different about the life of old people, and it's hard to distinguish the causes of the changes that you see because so many things are different. Do you see any way of investigating which are the important causal causes of the changes that you are seeing here?
Okay, you mean about the microbiome in this case?
Yes,
the fact that yes. Yes, of course, is a sort of combination of stuff. Yeah, there is not one single causes, and also sometimes it's difficult to understand if something ameliorate or affect the physiology of the host, or is the physiology of the host that change the condition, the gut, for changing the communities in some way. I think that is a sort of loop, a sort of cycle, a sort of loop where there is more inflammation in the intestine, in the host physiology, and so in some way there are the grew of some pathogens and inflammatory taxa is facilitated by this condition, and this in some way nurture inflammation. So this loop, we need to break this loop for ameliorate the condition, but there is not something that is clear connected as a cause-effect mechanism. Yeah.
And I guess there's a limit to what you can learn by studying mice in this context. You have to work with humans, and with humans, you have to take the data that come. You're not in a position to do manipulative experiments.
Of course, this could be a way, even if mice are quite quite different for several other aspects, but this could be a model for ameliorating the result and give new new theory or new hypothesis, for example. Yeah.
Thank you very much.
You're welcome. Thank you to you.
We have another question. Yes, Dr. Rampelli, why did you choose that specific kit for the shotgun sequencing?
Okay, there is not, there is more a practical reason above. So we tried this kit for preparing the library. We didn't have this time something for shade the DNA. And this kit include all the material that are necessary for preparing the library. We tried for the first time. We found that works well. At the end of the story, we repeat our experiment with the same kit. Just that.
Okay. Any other question? Well, I have a curiosity. I read in the past some studies where the main goal of the study was to know if using probiotics will enhance the cure of an illness or something like this, but not many related to aging. So, do you think that it could be a real interest on administrating this kind of probiotics to extend the life of people? Because I think that at least nowadays it's not something that it's in practice, I guess.
There are
not sure.
Yeah. There are several interests on that because healthy aging is well financed. Well, there are several also big pharma that are developing some probiotics on that. There are also some patents that are already known for Kristensenella and with the application for healthy aging, if I well remember. And so, well, is discussed now and for the future could be of interest for several aspects. Yeah, I suppose. Yeah.
Okay, because I'm also interested in, you said that we could or administer the probiotic itself, yeah, or we could use postbiotics.
Exactly.
So these are molecules produced by the
bacteria, such as short-chain fatty acid, for example, or something like that, that are the end product or some fermentation produced by the bacteria that the intestine eat and benefit from this molecule. So we can also, there are some medical strategy on that are at the first phase now. So, well, where they try to administrate this molecule instead of the bacteria. Well, sometimes is also better for a production reason. For example, for the production of Faecalibacterium prausnitzii, it's not so easy to obtain a huge quantity of bacteria, and it is also considered sometimes not safe to administrate the bacteria. So they prefer to choose this postbiotic way for be safer and for the administration also.
Okay, so it could be better in terms of toxicity or secondary effects, but maybe not as long-term as administrating the probiotics itself.
Exactly.
Okay.
This is the point.
Okay. So does anyone wants to ask any other question? Remember that you can use the QA box or raise your hand. I guess that there are no more questions. So if it's like this, I think that this is the end of this session. Thank you, really thank you, Simone, for this interesting talk. We could solve all the problems. You had good connectivity during the session. Thank you to all the participants also on behalf of NDPI. And this, as I told you in the beginning, this is the last live session of this conference. I hope you enjoyed the whole conference. You still have one this week to comment on all the submissions of the authors, and we are more than happy to receive any kind of feedback from anyone. So thank you.
Thank you.
Bye-bye.